Hello, welcome to Novo Talks. We now have an official title for the podcast. This is Amanda. I'm a senior technical support at Novogene, and in fewer than 100 days, I will have been here for 3 years.
Oh, really?
I have a countdown on my phone because that's when my retirement account estimate happens.
Oh, okay.
And with me is Harsh.
Hey, hi guys. I'm the director of technical support at Novogene. Thanks for tuning in if you are tuned in.
Yeah. And so today we are not doing what I thought we were going to do last time. Instead, we are going to be talking about another Novogene-powered publication, and we are going to try to give you guys sort of a case study-like summary of this one. So we haven't got a case study by our product specialist for this paper, but Harsh and I are going to try to do our best. So the title, just really quickly, is a 15-gene classifier to predict neoadjuvant chemotherapy responses in patients with stage 1B to 2B squamous cervical cancer.
That sounds so heavy when you say it like that. It's just like one of the heaviest titles.
It's a really big title, and basically these guys are trying to, statistically and using sequencing, of course, to give a very simplified way of just testing whether or not this neoadjuvant chemotherapy is going to be impactful for these cancer patients.
Correct.
Yeah. And I think that's mostly everything just to get us started that I had. I think you're going to take it away and tell us about cervical cancer, Harsh?
Yeah. So this was like an interesting rabbit hole that I went down. In your, what do you think, based on whatever you know, is like, give me the top 3 cancers that are responsible for like number of deaths. Top 3, top 4.
I was gonna say, I think it is in the introduction that cervical cancer is like the most predominant cancer among women, right?
It is, but it's if you just don't look at women, but just look at cancer in general.
Oh, like overall?
Overall, where do you think cervical cancer ranks?
I'm not sure. Probably not in like the top 5, right?
It's actually number 4.
Oh.
It's the 4th most common female cancer.
Yeah, right.
After breast, colorectal, and lung cancer.
Yeah. And I know that breast cancer is definitely like across genders.
Yeah.
Breast cancer is one of the highest and most deadly cancers we have.
Yeah. And I shouldn't say the funny thing, but just, we live day to day without even sort of noticing like what's happening in cancer research, but there are 600,000 new cases every day and 340,000 people die annually. Yeah. Just because of cervical cancer. And this is based on my understanding of how this progresses and what we can do to prevent it. We can prevent it quite easily.
Yeah. As someone with a cervix, there are a lot of good preventative measures, of course. On the one hand, in younger populations, preventing human papillomavirus is the best thing you can do. So they have a vaccine for that. Yeah. It's a few rounds, I believe.
Yeah, that's still in clinical trials, though.
Yeah, right.
That vaccine.
Well, they've been doing some different vaccines since I was a kid. Right?
Okay.
And then also annually, that's at least in the U.S., that's part of our annual exam by the physician. So you're also just double checking and examining that aspect as well. I believe though. So when I was very first being, I guess, tested for cervical cancer and having that screening done, it was being done annually, like at every visit that you did it. But I believe now it's recommended for certain age groups to not do it necessarily every year, right? So like once you move into like a higher risk age group, which I believe is on the like 35, 40 plus range, then I think you move to having that that testing done more regularly. But for maybe like 30 and under, I think they're testing it maybe every 2 years. cancers, but I
Yeah.
don't recall.
I don't. No,
Sorry,
I don't know.
that may be outside of what you looked up.
Yeah, I mean, I can't really contribute to that because I don't know.
Oh,
but
yeah,
that's definitely.
Yeah, the recommendation at some point, I think about when I was in college.
Yeah.
In the past, like 5 or 6 years.
So we've progressed.
Changed.
We've progressed, you would say, in terms of how fine-tuned the testing is and how much we've ruled out. You think?
I mean, what I know as far as it goes is that it is they're doing what I believe cell culture basically. So they're taking a sample and culturing it just to see
HPV.
And
Not HPV.
they are looking at for HPV as well. Usually that will be in like a blood test, but they are taking a swab of the cervical cells and just ensuring that there aren't any cancerous cells growing there on the surface, which is usually, based on what I read here, where it's going to start. It's going to be on that surface.
Yeah, makes sense.
So I guess we could assume that we've made some advances because we are. The recommendation is that we need to test certain groups less often. Right?
Exactly. That's what I was. Yeah.
So theoretically, we do have a better idea of who it's impacting, and maybe you're not needing to do the swab necessarily to.
Got it.
To know who needs to be tested.
Who needs to be? Yeah. Which is. But one other fact that was like just kind of like if we've got such advanced medical tech, and so it looks like it's the leading cause of like deaths, like cancer-related deaths in 36 countries.
Sorry, death as in not
like,
being alive anymore, or death as in owing money?
No, deaths.
Okay, sorry.
Death.
Yes, not being alive anymore.
Not being alive anymore.
Yes.
My death is I'm dying right now, basically. With as in.
Yes.
Can you understand though?
Yeah. No, I got you.
Yeah. It's like I know like my D's and T's and P's are kind of just the same.
Those words sound very similar.
Yeah.
Not your fault.
So,
yeah, right. So it has a high mortality rate. Is another way to say.
Yeah, it has a high mortality rate, but only in specific countries. We don't really see it in the US that much because obviously first world and things like that.
Well, I mean, also because, and maybe not to get too political, like Affordable Care Act and those sorts of things has made it such that anyone who does have health insurance, you don't pay for that exam anymore.
Yeah,
actually.
Yeah, so. It's just like basic amenities now.
Yes, right. It's the baseline basically for anyone who has health insurance at least.
Yeah.
So we are able. It's a lot more accessible.
Yeah.
Is what I would say is testing and catching it early on.
Yeah. So like in my opinion, not in my opinion, because I'm not like actually doing medical research on this, but I know a little bit. So I think definitely we have enough sort of understanding of molecular bio in this field to sort of make sure that we can prevent these numbers from surging and maybe even reduce that.
Yeah, right, exactly.
And so now one of the single most important sort of not important but the key factor in the onset of cervical cancer is HPV. I didn't know that.
Yes. Yeah, it's actually what I guess that's now they call it an STI.
Yeah.
They called it STDs when I was a kid, not to age myself.
Yeah.
But so it's a sexually transmitted illness.
Yeah.
And again, very preventable, basically.
Yeah, but one thing that I was trying to sort of research and understand is now when somebody's infected with HPV, is HPV known to sort of have it just mutates it's how HPV interacts with the skin, or is it just the general nature of HPV that's gonna basically cause havoc, like through and through inside, or is it just the interaction?
That's a good question. I don't know the answer to that off the top of my head. Exactly what about HPV is causing cervical cancer, right? So
I was looking into it.
Yeah, so obviously you can get HPV in other epidermal cells, right?
Yeah.
So,
Here's a thing though, like a little trend, which I obviously cannot validate, but I've just, based on research, it looks like it's a trend. So all these high level viruses like, HIV, HPV, they tend to make people more susceptible to cervical and breast cancer and all of that.
Okay.
So when I was trying to find out like what in HPV is resulting in cervical cancer, I came across some stats that patients with HIV also are kind of more susceptible to certain types of cancer.
Yeah.
So I guess if you're immunocompromised at some level, perhaps a mutation that is causing you to sort of not fight off that virus correctly is also triggering a reaction that might result in uncontrollable cell growth.
Yeah, and I think that makes sense with my cancer research knowledge, right? Part of there at least is an arm of cancer research that the goal is to figure out why our immune system isn't responding to cancerous cells correctly, right?
Yeah.
So I that makes sense to me in that respect, I guess.
Yeah.
That theoretically our immune system should be looking at cancer cells and saying there is at least something wrong, right? Not necessarily that it's foreign, but our immune system is a little bit more complicated than just being foreign versus ourselves. And yeah. So I wonder. Sorry.
No, go ahead.
I was going to just add to that. I wonder. So obviously, being immunocompromised, but if you is a factor. Clearly, that's a trend at the very least that we can recognize. So. On the other hand, you have some sort of autoimmune condition. I wonder if that also is something we could consider to be a factor in this. That's definitely outside of the scope of what you were looking at for this.
No, that's interesting.
That's another thing that just hit me in the back of the head.
Yeah, because in autoimmunity, your body just thinks that your own it all starts with your own antibodies considering your cells as antigens.
Right.
Exhibiting antigens which they shouldn't.
That your immune cells are looking at your cells and creating antibodies for those cells.
So what's causing that? So antibodies are thinking that this is an antigen even though it's not, which means that whatever factors or proteins that are on the surface that are supposed to say that hey, this is our own person, or this is our innate sort of biology, that's malfunctioned.
I will say my expertise in autoimmune, I couldn't even remember the word just now, so my expertise in autoimmune disorders is less.
No, that's fine. But it's kind of interesting that you bring that up because that recognition sort of potency, not potency, but that recognition success
of that recognition
is based on VDJ
important.
arrangements and how they are controlled by that's another rabbit hole to go down into.
Please don't take me with you. I took an immunology class in 2015, and
I had to take it in grad
at the
school.
Beginning of it, I thought I was going to fail. So.
No, this is a story for another day. But if somebody was to ask me what's my highest point in life, or one of the most biggest achievements, it's related to immunology.
Oh yeah.
But definitely. Yeah. But that's a story for another day.
Maybe we should get Jansen.
Yeah, he's an immunologist.
I was going to say he's an immunologist. Maybe we should see about him.
Yeah.
He lives in the area still, right?
Yeah,
Maybe
he does.
We should get him to come talk to us about immunology one of these days.
Yeah.
Everyone will love his voice. He has a lovely British accent. So.
Yes. So I think that's interesting.
That's a rabbit hole we could fall down another day though.
Yeah.
Right. So any other information about cervical cancer we should know going into this?
So that's really it. That HPV is the main reason. So right now we are at HPV is our sort of main causative agent, right? That's triggering the onset of cervical cancer. But if you have other infections, high level viruses like HIV and things like that, if you're infected with that, so that's when you know that hey, you might be in the ballpark of getting this.
Yes, right. That's that.
Yeah.
In these this certain environment, right?
Correct.
You are more susceptible.
Correct. And now what this paper is trying to focus on is treatment. How do we sort of manage this? Once we have it, how do we manage it? Because it is manageable and we can get rid of it from patients. But it depends on the stage, obviously.
And obviously, the cervix technically isn't a required organ. Obviously, you don't even have one, so it's not a required organ to survive.
It's to survive, okay. So
it's not required to survive. It's not like your lungs.
Yeah.
Right? And that'll get into sort of my next stage. Sorry, I'm preparing to segue myself in.
So right now, this might sound really obvious, but whatever stage you're at will determine, what sort of treatment you undergo. But mainly, the main thing that's being carried out right now is chemotherapy, and sadly, it's chemotherapy because we can't catch it in the earlier stages. So we just need to, be a little bit better and more disciplined in terms of, fine-tuning our diagnostic procedures to see, hey, this person can receive this treatment, this person cannot. And that's what this paper is actually trying to do, that, hey, we've come up with this novel way, and here's like a very, very highly vetted 15 set of genes that if we put that patient database through, we're like, hey, these 10, 15 patients will resist neoadjuvant therapy, or these 15, 20 will respond well.
Yeah.
So that's what's needed, and maybe if we have different sort of studies like this, we could collect all those and perhaps just have like a really sort of robust way of allocating treatment. So so I think this is a good point for you to sort of step in and sort of explain the novelty of this method.
Right. So there's a couple of words in the title that that I didn't know coming in. Right.
Yeah.
Number one is neoadjuvant chemotherapy. I am lucky enough not to have a ton of experience with what I now know. There are several different varieties of chemotherapy options out there for cancer treatment, which A, is great, and B, is also, the I don't know, benefit of living in 2022, I guess.
Yeah.
Right, there have been lots of great advancements in cancer research and cancer treatment. And the other part of this title that sort of caught my attention that I wasn't sure a lot of the details about was that stage 1B to 2B part, right? So if you have any experience with cancer, you know that there are sort of grades or stages, right? Stage 1 being the earliest and stage 4 usually being the latest survivable stage.
Yeah.
And so these researchers are specifically looking at patients with stage 1B to 2B. So each of those stages can have subcategories A, B, and C.
Okay, so now this maybe you know this, maybe we don't. But you said 1A to 2B. Sorry.
1B to 2B.
1B to 2B.
Yeah.
So how are these two different?
Yeah. Right. So 1B is described by the American Cancer Society as the cancer or the tumor itself having spread deeper than 5 millimeters from the surface of the cervix.
That's 1B.
That's 1B. So it's very, very early, right? Okay. So 1 being
less than 5 millimeters.
Less than 5. It's deeper than 5 millimeters.
That's still quite deep.
So yes,
you're talking micros, talking cells here.
The cervix is not that big. Either.
Yeah.
Right.
Yeah.
So 5 millimeters. Deeper from the surface of the cervix, all the way over to 2B, which is basically the cancer has grown beyond the cervix, and as well as the uterus. So the cervix is theoretically, I guess, or maybe anatomically, the opening to the uterus. Yeah. Maybe some folks might need to look up some anatomy before listening to this discussion, but so the cancer has grown beyond the cervix and the uterus, which again is attached to the cervix, and has started spreading to the nearby tissues that sort of hold those two organs together.
Now it's beyond the cervix.
Yes, so now it's beyond the cervix, and it's sort of impacting those nearby organs and the parametria. But it has not impacted the lymph nodes nearby. Okay. So I think that's an important distinction, that once cancer moves into the lymphatic system, it very readily can metastasize and move to the rest of the body.
Yeah.
Right? The lymphatic system is very similar to the circulatory system, basically, in that it connects everybody together.
Yeah.
And so I might posit that 1B is probably one of the earliest points that you would be diagnosed, honestly.
1B? Okay.
Yeah,
okay.
Right. I mean, if you're, again, especially if you're maybe going to your general practitioner or something maybe once a year, or maybe you're getting tested every 2 to 5 years, depending. Again, this is strictly in the U.S., and I know we were talking about this being you mentioned something and you said we, and I was thinking, maybe not we, because it sounds like the U.S. has things kind of handled on the front end.
Yeah.
Right?
Yeah.
So.
I meant like we as a society.
We as a society, of course.
Yeah,
but, we in the U.S., we are catching a lot of these things on the front end. But if you aren't, this might be one of the earliest points that a lot of folks are being diagnosed.
Mm-hmm.
I'm also not sure how the difference between 1A and 1B really, what your treatment options are at that point, I would say. So perhaps if the spread is more than 5 millimeters, maybe a surgical removal is just, or if the spread is less than 5 millimeters deep, maybe a surgical removal is possible.
Okay.
Sounds absolutely awful to me.
Yeah. Surgical removal of like just
of the cancerous tissue there,
tissue,
right?
Yeah. Mm-hmm.
So one, we have neoadjuvant chemotherapy. We're looking at very early stage patients. Okay. Basically. And neoadjuvant chemotherapy is different from the traditional, I would say, concurrent chemoradiation therapy. So they're calling it CCRT in this. Mm-hmm. In that neoadjuvant chemotherapy's main, or rather, it's not as effective for everyone. Okay. Basically. So that's why we are looking for this panel of genes to potentially vet who will respond and who won't. Yeah. Right? Mm-hmm. Whereas concurrent chemoradiation, as far as I understand, pretty much will
everyone.
Yeah. It's pretty readily responded to across the board.
Got it. But what's the disadvantage of concurrent chemotherapy?
Oh, the disadvantage is that it's very hard on the body.
Hard on the body. That's like general chemotherapy disadvantages.
Yes. Okay. Yeah. Right. So in general, chemotherapy disadvantages.
Yes. So how is neo or win basically is what I'm asking. In any way, what can be salvaged?
So neoadjuvant chemotherapy, basically, not everyone is going to respond to it, but it could.
Those that do,
right? Mm-hmm. Neoadjuvant chemotherapy could reduce the size of a tumor.
Okay, that's pretty good.
Right, and just overall reduce the size, and that sort of in conjunction with surgery, right? They're saying radical hysterectomy here, which I wrote down, is the surgical removal of the uterus, the cervix, as well as the ovaries and the lymph nodes.
That does not sound fun.
It doesn't sound fun, but like we mentioned earlier, you don't have half of those organs, so it's not necessarily a death sentence.
It's not a death sentence, but it's just if we can avoid
the surgical removal of any organs doesn't fun to me.
Exactly. So that's gonna be it's just gonna result in people making a lot of tough decisions in life.
Yes.
So if we can circumvent that, then why not?
Let me see. I was also just going to give a little bit of a note. So there's a handful of different neoadjuvant chemicals, basically, that we're able to take advantage of, I guess. Okay. And primarily, this research is looking at folks who have responded well to taxanes. Okay. As well as platinum-based chemicals. So
taxane and platinum-based chemicals.
Yeah.
What is taxane?
That's a good question, and I should have looked it up before.
Okay, we can look it up right now.
Let me make sure that I didn't write it down. I did not.
A taxane is a type of drug that blocks cell growth by stopping mitosis, cell division.
Sure.
Taxanes, that's a very high-level overview, but taxanes interfere with microtubules, cellular structures that help move chromosomes during mitosis. So it's deep. It's not like messing around.
I mean.
So, okay, so taxane is one neoadjuvant.
Yes.
Okay.
Right. The list that I found included 2-3 others. Okay. So it looks like it's about 5 different types. And there's sort of a group that are the platinum based ones. So in this study they are using paclitaxel, which is a taxane, and then cisplatin or cisplatin, which is the platinum one, aka Taxol, so Taxol, and DDP.
DDP. Okay.
Yeah.
What does platinum do? Like, is it like an...
Highly reactive platinum complexes bind and cross-link DNA. Yeah, so what I've got is that DNA cross-linking basically causes the DNA to connect to other strands, and similarly, it prevents the DNA from being replicated.
Okay. Oh, okay.
Yeah.
Okay, so it's blocking. Not blocking, but it's like inhibiting DNA cross-linking.
Yeah, it's sort of... So it's causing the cross-linking.
It's causing the cross-linking.
Mm-hmm.
Okay.
Yeah.
Okay.
So the DNA itself is responding to, I believe the exact word it used was active or reactive platinum molecules.
Okay.
So.
Okay.
Yeah. Yes.
All right.
Form highly reactive platinum complexes that bind and cross-link DNA.
Okay.
And it goes on to explain what DNA is, but I'm not going to do that.
No, that's fine. We can move on from this one.
Yeah.
Yeah.
So at the sort of offset, basically the goal here is to find folks who are going to respond to these chemicals, right? And again, specifically, they're just looking at those two. There are a handful of other neoadjuvant chemicals that are used. So that's, maybe we can come back to that as one of the disadvantages. But yeah, so they're looking at they're looking for folks who are responding well to that. That's not just causing like a stalling of the tumor progression, but actually reducing the size of the tumor in this case.
Which
and
makes the surgery less invasive, I guess.
Yes.
Hopefully.
Right, and it's going to increase the likelihood that they're going to basically remove all of the cancerous tissues. Right, because although 1B on the one hand is very localized to the cervix, once you get over to the 2B stage, you are talking about some of the tissues around.
Yeah.
And I personally haven't done any whole cavity surgeries into the human body, but I expect it's a little bit difficult to distinguish.
Yeah.
Especially like maybe the parametria, the sort of the tissues that are surrounding and connecting these organs together. It might be difficult to distinguish this.
Yeah.
I think that was pretty much what I have as far as this goes.
So can we sort of conclude that the advantage that neoadjuvant chemotherapy is offering is that we'll just reduce the size of the tumor if you respond to this type of therapy, and the win here is that the surgery would not be as invasive.
Yes. Yeah. So the win here is hopefully the surgery won't be as invasive.
Yeah.
And in some, I mean, at least in the U.S., right, some of the wait times, right, in the interim between while you're waiting for the opportunity to get surgery, maybe you're setting yourself up for better success as well.
Yeah.
Just kind of thinking logistically, I guess.
Yeah, and there was this interesting little tidbit mentioned in one of the sort of papers they had cited, was that if this works, I mean, if this, like whoever this works for, then they can actually undergo immunotherapy, perhaps you can harness your body's own T cells and B cells and sort of tweak them so that they kill the tumor once it's reduced.
So rather than surgery? Right?
Yeah, so you could potentially avoid surgery, but that's pie in the sky right now.
Right. That's a citation of this publication.
That's a citation. But you never know. Like it'll take thinking on these tracks to eventually.
No, and that's definitely, I know.
Yeah.
Right. I kind of pointed out that earlier, right?
Yeah.
That like our understanding of why the immune system does not respond and attack these cells is also an area that, sort of an arm of cancer research. And I hope that one day we'll be able to just harness that.
Yeah.
Rather than, introducing lots of chemicals to folks, as well as rather than surgically removing whole organ systems.
Yeah, I mean, cancer it's a massive undertaking because there are so many things involved, like, where you hail from, what's your race, which country, environmental factors. But I think a lot of research is being consolidated, and I think I'm nobody to say, we are just a part of that puzzle, like barely. But yeah, when you see papers like this, you know that people are trying to sort of, categorize different things, that hey, this is here, this is here, this is here. If not this, then this. If not this, then that.
Well, and I think maybe I might not have pointed it out, but these this neoadjuvant chemotherapy is not exclusive to cervical cancer either, right? So that's part of, and I think we'll get into it as well with sort of the genes that we found, that these researchers found, right? That a lot of this stuff is able to sort of cross over different cancer types, luckily. And that's also great news for folks who don't have cervixes, for instance.
It's just like here I believe they focused on cervical cancer here because I believe the research facility they operate at, they experience like a lot of patients. They are I shouldn't say welcomed by or but
so they are, a lot of these researchers are in gynecology, which is specifically this organ system basically. So yes, that's their sort of area of expertise as physicians as well as researchers.
So now you can see that physician mentality and sort of progressiveness play out here very clearly.
Yes.
So what they're trying to sort of eventually get at is that do we have like a patient bedside sort of screener that hey, if I have 5 patients, how many of these can be put through neoadjuvant therapy? Now for that they actually came up with like a library of 286 genes that were like
these a very long list of genes.
And that go ahead.
No, that was it. Well, they came up with several lists of genes actually using a couple of different methods, right?
And they eventually sort of narrowed it down to these 286 genes, and they're like, hey, this is a panel that might help us sort of, whittle down to, okay, if I put these patients through this sort of genetic panel, testing panel, then we know if they will respond to neoadjuvant therapy or not.
Right.
But they were, based on some biostats that they ran, they were able to sort of reduce this to just 15 core genes.
Which is great, and probably also something you as a person could sort of readily look at that with your human eyes rather than having to push it through a computer. Right?
So let's say if somebody like me who does not know as much as a gynecologist, and I'm like, hey, how do you know that I'm a good candidate for this therapy? And then you can be like, hey, you do screen well for this 15 gene panel. This gene does this gene does that, research backed, studies backed. So you are at least a little bit, pacified about.
I guess I was going to say the word mutation, that you left out the word mutation, but if we are thinking from a physician perspective, maybe saying the word mutation isn't the way to move. Right? But that is the basis for this, right? It's specifically looking at mutations of these genes.
Correct. You're looking at mutations of these genes. I completely skipped that part, but
again, if you're thinking from the physician's perspective though, and I wouldn't want to hear the word mutation if I was a patient.
That's but you're right. I mean, that's what they're looking at basically. Are any of these 15 mutated or not? Because these are known to be like driver genes here.
Right.
These are driving, like known to be driving, or like research back driving, like the.
Well, I was gonna say not all 15 have like the backing though, right? There's only a handful of them that we actually already have folks looking at with other types of cancer.
That is correct, but just based on, so here's the thing, it was kind of smart the way they did it. So it's like to sort of rather than researching like heavy all 15, you just, based on stats and how different protein interactions are taking place, you just narrow it down to elimination processes and be like, hey, these are the ones that I want to look at. But they did research like top 3 or 4 genes, which we'll cover, and they were kind of able to sort of assess that the rest of these 13 would also be in that, and there's some research to back it up.
And I mean, at the end of the day, from the patient and the physician perspective, it isn't necessarily imperative that we understand exactly what those genes are doing. We are just using them to say, you have what looks like if we're personalizing this, right? You yourself personally are going to very likely respond well to this treatment. Right?
It's just a screener. It's not like its function is not important here, but is it mutated or not is what's important.
Exactly.
If it is, you fit the bill. If you fit the bill, you don't fit the bill. If it's not mutated, you fit the bill, you can get neoadjuvant chemotherapy. So yeah, that's really it. So uh, so now we have these 15 core genes, and I don't know. So they stressed a little bit about one of these genes that belongs to the beta galactosidase synthesizing sort of bioprocess.
Yeah,
And
that's the ST6 gal2.
correct.
Sorry, I'm gonna say the gene names because I have them written down in front of me.
No, that's fine. I have it too, but yeah,
Yeah.
it's just yeah. Uh, it
For
sounds.
listeners, it won't be fun.
It won't be fun, but that's what genes are named, like CXCL3 and all that. So this one's
Yes.
called the ST6GAL2. Now, apparently, when I was trying to research a little bit about this gene, and I mean, the function's fairly simple. It's making like beta galactosidase and things
Right.
like that. But like when you start peeling the layers here, you will see that its core function here is that it is a pro-apoptotic cell. Now, for people who don't know what that means, like you can go ahead and explain apoptosis, I guess.
Oh yeah, no, I was saying
Yeah.
because cell
Yeah.
death. We want
Yeah.
so apoptosis is programmed cell death, right?
Correct.
So that's either the cell itself or the cells around it, sort
Correct.
of telling that cell it does not need to exist anymore for whatever reason.
Exactly.
Right.
That yeah, you're like, hey, I'm done growing. This is my limit, and
Yeah.
that's really when apoptosis kicks in. Like, hey, we don't need to grow anymore.
Mm-hmm.
So now this gene is supposed to be, a proponent of enabling that.
Yes.
Right? So now if this gene is mutated, guess what?
The cell keeps living.
The cell keeps living. Now
Even when it shouldn't.
even when it shouldn't. So that's like classic tumor, classic cancer.
I was gonna
trait.
say, right, that is what cancer inherently is the cell keeps living even when it shouldn't. So,
Yes.
Yeah.
Uh,
So that's if you're having that mutation, not only are you
But
rather-
the mutation is now here's the thing. It's a very specific type of mutation in this gene. So
Okay.
the mutation is hypermethylation. Only if it's hypermethylated, it'll its role is going to be completely sort of silenced. I believe. So people have sort of these researchers sort of conducted. They went like a little bit deeper, and then they knew that it's hypermethylated because of whole exome sequencing where we come in.
yes.
So,
yeah.
which is a good thing. So we'll get into that later. This is when I see this, you're like, hey, they wouldn't have known if we didn't step in here.
I mean,
So that's something cool, like a notch that we can sort of chalk up for ourselves here. So not that that's the goal or anything. Yeah, they are addressing a much bigger goal. But
also not me or you, by the way.
Yeah,
but
yeah.
someone on our team.
someone on our team. Yeah. Yeah. Then there's this other gene, I don't know if you want to talk about that one. It's called the POM 1-to-1 gene. Now, I did some. Now, it's supposed to encode like nucleoporins. Now, I know porin proteins are the ones that transfer sort of nutrients from like cell to cell, right?
Yeah.
So, and its mutation is known to aggravate prostate cancer. Now, I was just trying to think about it based on my own sort of little understanding of the subject here. That if there is a mutation in the gene that's transferring, that's transferring porin proteins. Now, that channel of porin proteins, perhaps it's not working properly, which means the cell is
Not taking or releasing,
yeah.
right?
Yeah, nutrients. But this gene, like, it's not validated for cervical cancer, but it's known to be, cervical cancer is going to be for females. You could consider the male analog to be prostate cancer, I guess. So it's supposed to aggravate prostate cancer.
Okay.
Now, I didn't do any research, but I can assert that it's basically the same thing. You're looking at, skin like layers below
Yes.
of tissue,
Right,
basically
right.
everything, right? So they also concluded that they've confirmed that this also plays
Yeah.
a big role.
Based on the data we have in this paper
Yeah.
specifically, it's also going to be impactful for cervical cancer.
Yeah.
Right.
But what's your take though? Like, how do you think would I can see, a cell that favors apoptosis, and if that's not work, or a gene that promotes apoptosis,
Right.
getting mutated.
That
and
one
that's a direct
seems
correlation.
very
obvious.
very obvious, very in your
What's
face.
with this one? How could this play out? Like
so
with nuclear poisons?
I'm not sure. The thing that I was thinking about, and you brought it sort of around to other cancer types, as far as POM121 being a mutated nucleopore, basically, right? Being a nucleoporin, sorry. Right, and as far as that not allowing certain nutrients, water, chemicals, whatever exactly it's allowing to pass through the cell membrane, my thought was it makes sense in terms of being resistant to this type of chemotherapy. Now, so for me, if we're using these chemicals to impact the DNA and impact cell replication, and we're expecting basically these chemicals to go into the cell, then if one of the methods of these chemicals, of these neoadjuvant chemicals getting into the cell to the DNA is blocked, then it makes sense that it would be resistant to this neoadjuvant chemotherapy. As far as its role in like cancer, like other types of cancer and just in cancer in general, not specifically the response here, I'm not sure. I wonder maybe, so are there not, let me try to go back to biology 1. I believe there are chemicals that other cells release to sort of trigger apoptosis, right?
Yeah, so generally speaking, I don't think neoadjuvants, based on what my general understanding, neoadjuvants are going to go through potent proteins to, impact.
Okay.
That. But they could because we don't know. So
I was gonna say you did probably more reading on this one than I did.
No, that's fine. But I think it's just I'm just wondering that if the lack of I'm just trying to sort of see that, okay, if you have like a mutation in a nucleoporin making gene, then that means that it's not gonna perhaps have some sort of substances go through that might promote apoptosis.
Yeah, that's my only that's the
only connection I can form.
Only
if
sensible, right?
Yeah.
But that's like so right in terms of response to neoadjuvant chemotherapy, it makes sense to me that maybe it's a venue for these chemicals to potentially enter the cell.
Correct.
But in terms of it causing cancer to happen in general, my guess would be very similar, that maybe it's blocking reaction, or it's blocking any sort of chemical interference from cells around it. That's, The beta-galactosidase gene definitely is a lot more obvious to me.
Can you say that last part again though? The one that you said before this one?
Sure, yeah. So thinking in the exact same line as the neoadjuvants, us trying to trigger the cell to die purposefully, potentially that it would be blocking other cells around it trying to trigger the cell to die, right? That it's keeping those different cell signaling from being able to enter the cell and cause it to respond to other cells telling it needs to die.
That's a possibility.
That's my only that's maybe a roundabout way that it's impacting prostate cancer and cervical cancer. But if we were looking for like a direct route, that's the only obvious one that I see.
I wonder if there's research being done on how do these apoptotic promoting if these are substances going through there, or like proteins or like compounds are being fed to the cell, or is it just based on recognition of some molecules exhibited on the cell surface that it recognizes, and they're like, hey, I'm done?
Yeah, that's outside of my realm of expertise for sure.
That would be an interesting sort of approach
question.
to look at it.
Maybe we should see if there's some research happening about that.
Yeah, because if it's just surface level, then that would be a different way to sort of tackle cell growth. Like,
Yes.
but if it's
Exactly.
something that's internal, that's taken place, that's resulting from like maybe excessive nutrients or deficiency of cell nutrients, that's a whole different approach.
Yeah, and that's right, and that would drastically impact, especially if it's maybe a certain cancer type specific. It may drastically impact how we are responding. Right.
Yeah, because those cells are different then for different organs. Yeah, that's interesting.
But that's another rabbit hole.
Yeah.
There's too many rabbit holes with this paper.
Yeah.
I personally am struggling to do the product specialist job, which is very briefly summarized.
No, I mean it's
to be honest.
Yeah.
Yeah.
But yeah, in general, I would say that these two were the main genes that I thought were good to discuss. The rest were, I think, they're also sort of known to be driver genes, which were narrowed down from like hundreds to these 15, and then the other 13 genes I didn't really look into them much because based on my understanding of the paper, they were just deduced to be driver genes based on pure statistics and elimination.
Yeah, right. And again, sort of to go back to what we said before.
Yeah.
In terms of a cancer patient and a physician, that is really all you need, right?
Yes.
And I think I may have left this part out earlier, but one of the advantages of this is that advantages, I should say, of having this panel and running this test before you decide to go down the neoadjuvant chemotherapy route is that it could save you a good 3 to 6 months.
Yeah. Yeah.
And that's a
time savings. Yeah.
At the end of the day, 3 to 6 months makes a big difference between stage 1B and stage 2B.
Yeah.
Of any or stage 1 to stage 2 of any type of cancer.
Yeah.
And so right, it is of interest to us, of course, to know what these genes do and how they are playing a role in cancer. And it is definitely interesting for researchers who have other areas of interest than dealing with patients, which I believe that these researchers, that's their main focus, is trying to figure out solutions for their patients.
Yeah. So what did we do here?
So basically what Novogene did was the whole exome sequencing part of this.
Yeah.
I believe that we also did the analysis, considering we have a handful of our team actually as authors on this one.
That's pretty cool.
So I suspect that we did the analysis. But basically we were doing the whole exome sequencing. The test group of patients was on the smaller side.
Okay.
Right? And I think we talked about this off recording, but so they do have a smaller test group of 56 patients that we used for the whole exome sequencing for just figuring out, again, statistically, those 15 genes that we came to that conclusion, right? But really the 200, almost 300 genes that we started with, right? So that whole exome sequencing on tumor and regular tissue samples, I believe, of those 56 patients. We were doing that whole exome sequencing aspect and then basically output this long list of genes that they narrowed down. Obviously, 56 patients to set up a gene panel is a little bit on the limited side, but of those 56, they were sort of in a handful of different groups, and so we do have like the ability to do some comparisons between them, right? We're not just looking at 56 who were successful. We're looking at 56 patients, let me see, 26 of whom had a response to the neoadjuvant chemotherapy.
26 out of the 56?
Yes.
Like positive?
So, yes, who had basically a positive response being either, so one patient had a complete response, which was the tumor disappeared from the neoadjuvant chemotherapy. 25 had a partial response, which is more than 50% of the tumor size decrease.
And these are from the same group? these patients?
These are from, so they have something like 1,000 patients in a data bank that they were looking at, who they had access to tissues, basically. And then of those 56 who actually underwent the neoadjuvant chemotherapy.
Yeah.
So we have 26 who had some sort of response.
Got it.
Basically.
And
so
then
some
1
sort
versus
of
25.
One of those completely, the neoadjuvant chemotherapy was very well received, basically. So one of them had the tumor basically effectively disappeared.
Got it.
Whereas 25 of them had something in the realm of like a 50% decrease in the size.
I was just trying to sort of see the reason I asked you that. Was there a difference in the group of this one person and the 25?
No.
Same
So
group?
there's those two. So they are putting them into 4 different groups. So they have complete response versus partial response.
Okay, so it's post-response.
Yes.
The groups are post-response.
The complete and partial responses were both considered to be within the group of people who were responsive to the neoadjuvant.
Makes sense.
So they have 4 different groups, 4 different categories for the patients, but they are sort of breaking them down into 2 groups. So the other 2 groups are stable disease. So after undergoing that neoadjuvant chemotherapy, the tumor remained the same size.
Got it.
And then also, so that's the majority, by the way. So there's 30 patients left.
Okay.
Right, we have 26 who were responsive, we have 30 who weren't. So 29 of those maintained the tumor size over the course of that therapy, and then 1 actually was so not responsive that the tumor grew.
So out of the 56, so the 56 were kind of like they will be impacted by, they could be positively impacted by neoadjuvant, or was it just?
So this is the front end of creating the gene panel.
Got it.
Right, so we have 56 patients who underwent that chemotherapy treatment.
Got it. So this is what led to the gene panel.
Yes.
Got it. Okay. Makes sense.
So that's they just have these 2 categories of patients. So those who are responsive.
This is what's resulting in the hypotheses and not testing the hypotheses.
Yes. Sorry, this is not the test group. They do have a whole other group of patients that they did use the panel on.
Same page now.
Yeah, that they verified the panel with.
Got it. Yeah.
But that's basically, so they did have sort of a somewhat even split, 26 and 30 responsive versus non-responsive patients. And those are the patients whose data we used basically to do that whole exome sequencing to produce that 200 some odd genes of interest, right? Or rather mutations of interest, really.
Okay.
Right? And then they had patients come in, so they had 205 patients between 2016 and 2018. They reduced that down to 102.
Okay.
Right? So 103 of them they had to exclude for a couple of different reasons. But they had new patients come in. So that original 56 is from a data bank. They have new patients coming in to validate the 15 gene classifier after that.
Okay.
They were a little bit, they used this 15 gene classifier to sort of determine and push these folks, based on the results, they pushed these folks to do the neoadjuvant chemotherapy.
Okay.
And we're looking at 22 out of those 102 who were considered not responsive based on the original criteria of either the tumor remains the same size or it grows.
Got it.
So the success rate is, not 100%, of course.
Yeah.
Basically of testing whether or not
this works.
This 15 gene panel is a good,
it's reliable way of.
It's reliable, right?
Mm-hmm. Would you say it's reliable?
I would say, in terms of, right, exactly what we wrote in my notes was play good cop and bad cop. And sort of discuss the disadvantages of this study. And while from a researcher exclusive perspective, I would say I probably wouldn't find this to be sufficient. Basically, as a publication, as the data set as a whole, right? We don't have enough patients on the front end to even be making broad statements about these 15 genes, in my opinion. 56 patients is not enough.
Yeah.
Right?
And they sort of acknowledge that.
They
like,
do acknowledge this.
Like
yes.
completely, like
Yes,
in
yes.
straight terms.
Right? And
I
100.
believe they will be doing more research on this.
Oh, for sure, right?
Yeah.
And again, too, like 100 patients for doing this testing is also in terms of the grand scheme of research community, not enough.
Yeah.
However, this is a cancer research paper, and if I were a cancer patient, I would not be upset that this was published, right? So thinking about it from the two different perspectives, right? And if I were a physician, I would not be upset to just have this information in my back pocket and publish it as soon as possible, as soon as you have something, right?
Yeah, yeah. There's one thing that they could do to reduce the work here. I was thinking about it.
Okay.
Like if they had to make it more comprehensive.
Yeah.
Then this is just, I don't know if this, I think this should work. So you have like a database on like, GSEA.
Yeah, right.
And like things like that. You have Geo and all of these. You could plug in these these 15 genes and see how they did in from different parts of the world studies on cervical cancer.
Yeah,
If
right.
there is
And
like
or
that.
other cancer types
Or other cancer
as well,
types.
right?
You could do that and see how.
Yes.
I'm sure that has crossed their mind.
Yeah.
I'm sure.
Yes,
So,
right. And that's exactly like, again, thinking about it from a researcher perspective, that would be something that would clinch this for me, right?
That
would feel more substantive
Yeah.
as far as making grandiose statements about
Yeah.
this. Okay, so we have this gene classifier,
Yeah.
right?
Yeah. I
At
see.
an 80% success rate, that's great,
Yeah.
and it is great, right? From a physician's perspective and from a patient's perspective. But you still do also have 20% of those patients who are wasting 3 to 6 months and
Yeah.
losing time.
Yeah. That's.
And again, like from the other hand, that may honestly be outside of the scope of their capabilities as researchers too. It would be outside of the scope of mine.
Yeah.
I would have to find a collaborator if I were leading this type of a study. It would be outside of my scope. It would be something that I would be comfortable or feel confident doing.
Yeah, but you'll need that.
Sorry?
You'll need that.
Yes.
Yeah.
Exactly. And that's so. I have mixed feelings about it being published.
Yeah.
I'm not sure if that's a controversial or okay thing to say as a Novogene employee and this being a Novogene-powered paper.
Yeah.
But in terms of statistical significance, I don't think that they've got enough patients. I don't think that, like you said, there are banks of research and banks of data, like whole exome sequenced samples for globally. There is a data bank somewhere that exists that has that data for them.
Yeah.
Obviously, again,
Is there though? I know, like from my brief interaction with GSEA and like submissions on Geo, that you could perhaps do it.
You could perhaps pull something together at the very least.
Yeah, and if you just search genes in general, like even on NIH's website, you do get like publications associated with that particular gene.
Yeah.
Yeah, so if you enter these 15 genes, you will see.
At the very least, it would be, I guess
US-based
maybe
studies
that would not give you information about response to the neoadjuvant chemotherapy, right?
That would not.
And I think that's also part of the reason why the scope is so small, right? They're using a data bank specifically from, within their reach, basically, and out of more than 1,000 patients, they found 56. So, maybe
you would need collaborators.
You would need
in
some
different
sort of collaborators in different parts of the world, and I'm not sure if a large database would even have data on whether or not those patients had undergone
certain
chemotherapies before or after. It's a very niche area of research. So
you know what? You will need You'll need to do the wet lab here. You can't do it through the database.
Yeah.
Because that database is going to be so niche that you're basically hoping for that study to exist.
I think at the very least it would be interesting to see
just their profile, these genes are.
And also even just how that gene classifier, what it looks like in terms of other types of cancer. Right? And just to see what is out there as far as that goes. I think that would beef this paper up
Yeah, absolutely.
Basically,
Yeah.
from again, strictly from a researcher's perspective.
Yeah, but I would say that uh, I would kind of take, since we should look at the negatives and the positives. So I'll look at the positive.
Oh, yeah.
You gotta start somewhere. So
Yes.
this is a start somewhere, and I think
Absolutely.
the fact that you and I are thinking and talking about it so much means that
They did something.
they
right,
did something right, and I think it's okay for a publication, but I do see your perspective completely. But about it's that whole thing that sometimes researchers and hospitals, it's a sad fact of life, but it's true that they are pressured and judged by how many publications they dole out,
Yes.
right?
Right.
It's that's just how
Yeah. I
how
mean,
our academic society has sort of shaped up to be.
I was gonna
Sadly.
say, yeah. I can't speak to
Yeah.
these folks. They are based out of China, but
Yeah.
my university and my department at my university was very closely affiliated with the USDA, and they
Yeah.
even like the U.S. government has minimums as far as you need to publish this many papers annually,
Yeah.
regardless.
Yeah, that's just
you know.
to keep somebody. I mean, there are good things about it, and there are bad
Yes.
things that
Yes.
sometimes you get stuff that is like, hey, that should just be contained within a conversation, and
Right.
sometimes, and the other side is that hey, that's motivating for people. That makes sure that you're not complacent, you're doing stuff because the government is paying you taxpayer money for conducting research.
Yes, right,
So,
and it's definitely
sense.
one of those double-edged swords, though, that
Yeah.
if you are pushing people, if you're pushing maybe the wrong type of person,
Yeah.
right? They may
Yeah.
just publish what they can publish
Yeah.
for the sake of
Yeah.
doing their job.
But I can say wholeheartedly that this falls in the good category of
Yes.
that double-edged sword.
Yes, for sure, and that's like, if again, not to keep bringing it back to being the patient, but like if I was a patient and they said, okay, we have this publication, they started with only 56 people, like as a researcher myself, I would say I would just do the gene panel, whatever,
Yeah.
right? If it were me.
Yeah.
And knowing what I know, I would say, great, let's just see what it says.
Got it. Mm-hmm.
So
Yeah.
to do a little good cop, bad cop,
Yeah. Yeah, makes sense.
it's and trying to maintain that,
Mhm.
these are human people on the other end of things, I
Okay.
guess.
So overall, I think, do you agree that we can sort of give this one like a win in terms of moving the needle forward a little bit?
For sure. Yeah.
For
yeah,
cervical
yeah.
cancer?
For sure. And that's like basically all of the folks that they cited who are doing other types of cancer research
Yeah.
need to now look at this group of genes.
Yeah, they should.
A.
Yeah, I would.
I
So,
would if it were
yeah.
me.
Yeah.
Yeah, but that's I think it's great that Novogene was able to be a part of something like this.
Yeah.
And this maybe will segue into my little pet project or my little area of interest, which is personalized medicine.
Yeah.
Right? I do think that this is also a great step in the direction of personalizing medical care for everyone. On the other hand, these researchers are based in China, and I don't know that this would be sufficient for someone to receive this kind of a treatment in the U.S. for
It
instance.
wouldn't.
I mean, I believe wholeheartedly that it wouldn't.
Even in China, it wouldn't.
Yeah,
Yeah.
like it.
They say it.
Yeah.
They address it. They're like, hey, we'll do more stuff, and this is like it's like a subheading in the paper. The limitation of the study is bear in mind that it's only a starter. Like we're
Yes.
just gonna explore more if we can. But if you're looking at just personalized medicine in the Chinese population, maybe it's a start. You don't
Mhm.
know. So
Yeah, and I'm just thinking from like
yeah,
what I know about the FDA in
Yeah.
general.
yeah,
Getting something like this
hmm
to be sort of backed by the FDA for one, and
Yeah.
also be covered by health insurance,
Mhm.
might be a bit of a logistical nightmare, basically.
yeah. I mean, this is definitely gonna take for this to sort of for personalized medicine in general to
Yeah.
reach like a scale of just a regular medical treatment, or
Yeah,
just to be
yeah.
considered as regular medical treatment, like as you go to the doctor,
Mhm.
simple copay and all of that. It's gonna take like a revolution. So
I mean,
not a revolution,
and
but
a lot of data.
yeah, and a lot of data to sort of convince.
Yeah.
I won't go too deep, but yeah, I know for a fact that by the time a drug is sold in the US, it's basically very, very expensive. All right, so
Yeah.
you would chalk this up as a win then?
I would chalk this up as a win. I would chalk it up as a good step in cancer research, but also a good step in the direction of personalized medicine.
Okay. Any other thoughts you might have on personalized medicine, or do you think, or should we just leave it at? I think we can say that this is a good start.
Yeah. I think that it's a good start. I think that it is. How to say? So we did whole exome sequencing for this, right?
Yeah.
And is whole exome sequencing maybe too much to do, basically?
For this one?
No. So in terms of personalized medicine in general, maybe that's the only other segue that I or that's the only avenue that I was sort of thinking is like, would this be more
Target region sequencing.
Target region sequencing. If I was to if you were Pipe Dream to release this as a test today,
Yeah.
I think doing whole exome sequencing might be a little much. But on the other hand,
Yeah.
target region sequencing is significantly more expensive
Yeah, because
and time consuming.
yeah, it's just because a lot of people are not. It's more cost. Is that correct? It's cost effective.
Oh yes,
It's cost effective to go whole exome
way
because it's
more
just
cost-effective.
more ubiquitous.
Yeah.
So the reagents and stuff available, you have to spend more time on the back end,
Yes.
but it's easier to get data. Like you can devote like less time with bioinformatics to get to where you want to get to in terms of
Yes.
genes, rather than just hone in the wet lab, which is like design reagents to get those 15-20 genes you want to look at.
But then,
Uh-huh.
if we were thinking from a data security perspective,
Yeah.
would you want, like,
What?
it basically at the end of the day, if you were doing whole exome sequencing for a personalized medicine strategy,
Yeah.
but you are only specifically interested in these 15 genes,
Yeah.
you might be concerned about where the rest of that data goes and who's looking at it.
My take on that is a little bit different.
Oh
Like,
yeah,
that's
23andMe has my data. It's fine.
Yeah,
You don't have
that
to.
that is.
I'm devil's advocating over here.
Yeah. I mean, it's.
I'm fine for 23andMe to
If
have my
it's-
data.
yeah.
I'm lucky enough to not have.
Yeah, if I can just
Interesting
blindly
stuff.
walk into that illusion that hey, these people are good and they will
Yes.
eventually just look at their researchers and they want to help the world, and if they're going to look at these 15-20 genes only, even if they have my entire exome data, then I'm not worried.
Yeah.
Yeah. But that's a different conversation, I guess.
Right, and that's but the problem is to further personalized medicine.
The reason.
We do have to. Some people do have to say, okay, you can have the rest of it too.
Yeah, I'm curious though. Like, is there a particular experience that you've had that made you think like, where's that data going?
Oh, the experience was when Google sent me an email telling me that, hey, do you want to look at all the places you've been for the past like 3 years?
Okay,
And they showed me all of the stops that I took on a
Yeah.
trip to Germany in 2015.
No, that's true. That I mean, that
But,
I
like,
understand.
But
This side, yeah.
but my DNA, basically.
Okay.
And I mean, big data is big data,
Yeah.
right?
Yeah.
But I think of it, Google having kept track of all the places that I visited in Germany in 2015 felt really weird and invasive
Yeah.
for a second there.
Yeah.
And I know we talked about my love of Gattaca, the
Yeah.
movie.
Yeah.
So that is also in that same vein of the
I see.
slightly dystopian future
Sure.
of everybody's genetic information being out there and it being potentially used for discrimination purposes, for instance.
Got it. I didn't.
That's
That's
the
an
plot
interesting
of
point.
the movie, basically.
Yeah. It's a brilliant
I'm done.
movie.
That's
Yeah. It's one of my favorite movies.
one of my favorite movies.
Yeah.
10 out of 10 recommend.
Yeah. Even Predestination's pretty good.
Yeah.
I ended up seeing that.
Oh yeah, you watched it.
Yeah, it's pretty good. Pretty solid.
It
It's
was pretty crazy.
It's a mind-bending loop
Yeah.
that she gets into later. Anyways,
Yeah.
I don't want to give it too much.
I know.
It's a good movie.
It was a good movie. That's
Yeah.
all. Yeah. Ethan Hawke was
Just
also in that movie.
very
That's the only reason.
underrated actor.
Yeah.
He has a new movie that's called Black Phone. Apparently, it's 7.98 on IMDb, which is like a good thing.
I think his daughter is in Stranger Things, which is really popular right now.
Okay. I guess that's good.
Maya Hawke.
Okay.
Anyway.
Interesting.
Not to go off too far off on a tangent about actors, but yeah, that's and so, you know, when you're thinking about personalized medicine, there's a lot of all of these different pieces, right? It's like obviously FDA regulation, but then on the other hand, data security.
Which companies, except for these, I shouldn't say jokesters or whatever, except for like 23andMe and Ancestry and all that, is there are a few companies that are completely core focused on diseases, and they operate within the realm of FDA approvals.
Yes. 23andMe is not one of them.
It's not those. Like what are
historically?
Do you know? Like,
off the top of my head, I don't. But I do think that again, not to be too U.S. focused, but I do think I think you're right. I think there are a couple of companies that are FDA approved as far as that personalized medicine, disease and diagnostics.
Synapse is one. Fabric Genomics is the other one. Is what comes up. But like all these, the funny situation is, you know, it's so society is like so sort of bent these days towards, you know, making money. The first search is like top personalized medicine companies. Now, it's so funny that, you know, rather than this is just Google, I guess, Google search engine, but the first sort of search results that come up are basically just stock focused. Like, you know, that these are the ones. But I think so. That's how it charts it. Top 8 precision companies in the US, Industry Wired, Venture 8, Pharmaceutical.
I have not. I know I talked about my 401k earlier, but I don't know what it's invested in, so
now apparently,
I'm outside of that.
searchinglifescience.com has this article that sounds kind of meaningful. It says 5 companies paving the way for personalized care. So there's Precision, Quartz Bio, Congenica, I've heard of Congenica, Cited, CYTED, Multiple AI, Synapse is the other one. And we can get into, you know, we should do one whole situation on personalized medicine. That'll be fun.
I mean, this was the
that can be the special towards the end of
this
a
is
quarter.
This was me putting my toe and putting my hat in the ring, I guess, and suggesting that we delve into that another rabbit hole that we found that I could at least pique your interest in.
Yeah. But what's our fun one for today, though?
Yes, right. So last time we had a video that Harsh showed me of a roof.
Yeah.
The personification of a roof. That was meant to sell you.
It's a great ad.
It was a good ad. I did get some feedback that having us watch a video over a sound-only platform didn't work for some folks.
Didn't work, I can see that.
So today I brought another special interest piece from my side. So I did my undergrad and grad school at Mississippi State University. We are an SEC, so Southeastern Conference for American Football school. You might have heard of Dak Prescott. He plays for the Dallas Cowboys these days.
Okay.
I was there when he was there. Anywho, one of the areas of research interest in some of our departments was improving, or rather maybe decreasing the amount of concussions happening with American football.
Good luck with that.
Yeah. So basically improving helmets to reduce concussions.
Got it.
And one of the...
Oh yeah, helmets do exist.
Yes.
And still.
Yes.
Oh my god.
And still, the amount of concussions, if you don't know off the top of your head, I don't know the exact number, but the amount of concussions that happen in American football is deadly, actually.
Let's put a number on that.
Put a number on it.
Concussions. See, how do you spell C-U-S-S-I-O-N-S?
S-S-I-O-N-S? Yeah. So before Harsh finds his number, one of the, what's the right way to say? One of the areas or the avenues that was being taken was woodpeckers, because woodpeckers constantly slam their head up against something very solid. And the thought was maybe their skulls have some sort of internal situation that we could use to also improve the insides of helmets.
Our brains are already small.
We got a number?
No, I mean, I was going to make a joke saying that.
Oh.
Yeah. We are small. Like, so the number, okay, guess. Guess the number of concussions that result from NFL.
Just NFL,
Just
by the
NFL,
way. The amount
just
of high
NFL.
school football
Per year.
concussions
Per year.
is per year in the NFL. I'm going to say 1,000.
250, 247, 250.
250?
Yeah.
Oh, that's not as bad as I thought it was.
I mean, still.
It's still also only the NFL.
Yeah.
So.
Yeah. It's still the NFL, exactly.
Like,
so.
That's not including college level teams. That's not including rec teams. That's not including high school and middle school and elementary school. You know, they put kids in football in elementary school, American football.
I'm not
Where
surprised.
I'm from.
Yeah, I'm not surprised. It's
teeny tiny little helmets and shoulder pads.
Yeah, they're cute though.
It's cute, yeah.
Yeah.
I don't know the rate of concussions among elementary school age children, but
yeah.
Yes. So the thought process here is maybe the woodpecker has some sort of cushioning happening in its brain.
So was the woodpecker selected based on whatever you read about this?
Mm-hmm.
Was the woodpecker only selected because it's hammering wood?
What do you mean?
As in because we perceive that its head's gonna take like a battering?
Yes.
Okay, so go ahead.
Yes,
Go ahead.
specifically
Okay.
because
Okay.
no one had a ton of information about the physics of the situation, and that's basically what this paper is discussing, is the actual bare bones physics of the situation.
That's pretty solid.
So that's an area of research that was being talked about a ton when I was an undergrad, and it turns out that woodpeckers don't actually absorb the shock of hitting. So there's not any sort of cushioning happening inside of their skull. There's not anything special happening. They're just taking the full brunt of it.
Oh, okay.
Because if they actually were absorbing it, and this is the physics part, they wouldn't be effective at pecking the wood.
Okay.
So in terms of American football, it kind of sounds like maybe if we weren't effectively absorbing the regular physical hitting of another person, it may not be as impactful, actually. Basically, what they found, and I printed out this several pages just to give you the brief summary, what they found is the woodpecker's brain is actually just small enough that it doesn't matter.
Oh,
Yeah.
that's the conclusion?
That's the conclusion.
That it won't be concussed.
Yes. That the scale
or the
of
woodpeckers
their
like
brains,
perpetually concussed.
No, the woodpeckers are not concussed at all. This
Okay.
is it's just the size of their brains, their skull,
Yeah.
the situation they find themselves in.
Yeah.
As far as that goes, they just aren't getting a concussion at all.
That's good.
It's good for the woodpeckers.
Yeah,
I
yeah.
guess.
Yeah.
It's not great for helmet research for American football.
It's not really.
But yeah. The primate brain is just too big.
Yeah.
That's the point.
They should have looked at maybe like a bull or a cow skull, like a bull, like the bulls that.
Yeah,
race.
I think that was outside the scope for these folks.
Okay.
But
yeah.
But yeah, right. Like
I would think
there are definitely other animals that
they can head-on impacts.
Yeah, that take head-on impact, like rams and bulls
bulls.
and some different types of goats too,
Yeah.
right? That just ram their heads into things.
Yeah.
I think the woodpecker, just because it's so like
small and
prevalent,
Got
is
it.
probably a lot easier to rear and keep
Got it.
and research, but also just like it's ramming its head against things is constant.
It's
right?
constant,
It's
yeah.
going bam, 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 which is definitely going to be overkill in terms of a football helmet, hopefully.
Yeah, yeah.
Right?
yeah.
They're usually doing a one-off, maybe.
But if you're going all the way, then just look at
But
the
if you're
woodpecker.
going all the way,
just look
a
at that.
woodpecker makes sense.
Yeah, that's how you do. That's the safety
so-
test. Extreme
Yes.
safety test.
So unfortunately for these guys, it doesn't sound like the woodpecker is going to be the avenue for improving helmets. I said this earlier when we weren't recording, but maybe we should just cancel American football.
And how woodpeckers play football?
Maybe, I guess, right?
You still get the thrill of the
it's so
sport.
incredibly dangerous.
For the woodpeckers? No. It's not dangerous for the woodpecker.
Well, it's okay.
You never know, like how a
never
woodpecker.
know.
that's-
Their brains are very specialized. Their whole setup, their skull and everything is set up specifically for them to be woodpeckers, and that's it.
Yeah.
And American brains and American, like, human brains and human skulls were not set up to be football players.
Yeah. I don't know if this is like
so that's
too
my
much.
that's my little fun.
No, that's good though. But
Yeah.
if you, okay, so let's end the fun one on this.
Sure.
If you had to realistically pick an animal to sort of learn from that, hey, their skulls perhaps can take like a battering. Let's invest like a little bit of genuine money in this research. Now that woodpecker is like history because we didn't really reach anything conclusive. But if you were to truly advance helmet technology and learn from an animal based on its habitat and habits, I guess, which animal would you pick?
I probably don't have enough knowledge to make like a really good science-based answer. Just
No,
because
but whatever
I don't know.
knowledge you have from Discovery
Yes,
Channel, Animal
yeah,
Planet, or whatever, like just based
But-
on that.
Brain size relative to skull size is something I don't know a ton about.
No, but if
I
you-
probably would pick like a ram or something like that.
Like a ram?
Yeah.
Okay, yeah. I would pick
I'd be interested to see maybe how the horns
Yeah,
have an impact.
yeah.
Maybe
I've
there's
seen-
some sort of very basic physics knowledge here. Maybe there's some sort of distribution happening
Yeah,
with like widening.
yeah. I would also, so here's one thing that I was thinking. If I had to pick, I would pick one of those Spanish bullfight bulls.
Yeah.
Because I've seen them, so the horns do take some, but I've seen some without horns, they're like very small horns. They can topple cars with their head.
Oh my gosh.
Like they
mean-
just go full speed.
I don't know if they're getting concussed.
Yeah.
Either.
But I guess, would you say that the size of the skull is directly proportional to the chances of getting, or inversely proportional to the chance of getting concussed?
I
Could be,
think
right?
that it's just the relationship between the size of the brain and the size of the skull more than anything else.
So
right?
what is a
So part
concussion
of
medically?
Concussion is your brain hitting the front of your skull and bleeding.
Bleeding.
Yeah, like it's causing physical damage. It's breaking cells open and it's causing bleeding to happen. Right, like
That's a
so
concussion.
It's like a bruise.
Mm-hmm.
That's what a concussion really
So your brain is bleeding.
physically is.
So your brain's bleeding.
So if you have space, your brain can sort of jostle around without hitting the bone.
Wow.
That's what I gleaned from
Hmm.
this. But yeah, that's my understanding of what a concussion is.
Got it.
Also, I'm not gonna speak as an expert on
Yeah.
concussions either. I've never had one.
Yeah.
Because I play it very safe with my head.
Yeah.
I guess.
That's interesting.
Yeah.
I think this one's a wrap up then.
Yeah. For quickly, the paper I was referring to is called Woodpeckers Minimize Cranial Absorption of Shocks.
Oh,
But
okay.
again, it sounds like that's going to be promising. It's not. I went on Science Daily and they gave me a really nice summary of this.
Oh, no, that's,
And they were basically like, yeah, we're going to have to find something else,
yeah.
somebody else to look at.
We'll link everything here in the description, right?
Yeah, we'll
So-
have the paper, the woodpecker paper, as well as the cervical cancer research paper
Got it.
linked. They're both very readily accessible. I was able to print them out and didn't even have to go on like Sci-Hub or anything
Oh, okay.
sketchy.
So yeah.
Thank you for listening.
Yeah, thanks for listening, and hopefully this got you like at least a little bit of got you to get interested in cervical cancer.
Yeah.
If you're a science person, perhaps if you're researching cervical cancer, this might help you a little
Or
bit.
if you research other types of cancer
Yeah.
and you were like, wow, I didn't know Novogene was that cool.
Yeah, that's
Novogene's actually really cool.
yeah. We work with a lot of things like this, and it's
Yeah.
just you learn every day, and just these conversations can sort of perhaps help us and someone else like maybe develop or like get to a breakthrough. You
Mhm.
never know.
Yeah.
And even for woodpeckers, I guess. So
Even for woodpeckers and American football players and
yeah,
everybody's impacted by science every day, something like
That's-
that.
I want to also see the concussions in rugby.
Yeah,
That's without a helmet, by the way.
very, very US centric here.
Yeah,
Um,
the U.S. has
I
a
know.
rugby team.
yeah,
They're
yeah,
pretty good at rugby.
but
Yeah,
football, American
Football,
football
yeah.
is our,
it's your thing. Yeah,
it's our thing.
yeah. So much money in the NFL though. It's
Yeah.
a crazy amount of money. Yeah.
Did you also, just a side note, did you
Yeah.
know that football used to
Okay.
be worse So now football also can involve like a lot of passing and throwing,
Yeah.
right? But the amount of time spent like physically just wrestling each other and running into each other used to be even more.
That's fun though.
It's not. It's concussion.
No, I
That's
know.
what it is.
I've played like with the gear once.
Oh wow.
And yeah, it's just like
It's a
it
lot
was
of
fun
gear
banging
also.
into your friends.
Yeah.
It's just like
It's just fun
it's
running into each
your
other.
friends. Yeah.
Isn't that what like Red Rover is for?
Huh.
That's a
Red
game.
Rover.
It's a
Oh.
kid's game.
Oh, okay.
You all stand in a line and you lock hands. And then it's Red Rover, send so and so,
Mm-hmm.
so whoever on over, and you try to keep them from breaking through the line of kids.
Oh, okay. We have
But
one.
you're not hitting your heads against anything.
Yeah. We have one like in our boarding school, and maybe this is just like a Bombay thing, I don't know.
Mm-hmm.
In Bombay also my school had it. So you, like guys line up against a wall like this. So like making a train.
Okay.
Like that. Last person in the line has to run and try to jump over everyone
Oh
and
yeah.
land on the first person.
Yeah.
Like who the closest you can get.
Yeah.
And then the last person does the same thing. And then it keeps going. It's just like a fun game. Yeah. But anyway, so thanks for listening again, Amanda. Thanks for hosting. And yeah, hopefully we can kick out another sort of interesting topic for you guys in the next session. Bye for now.
Yeah, I was gonna say I don't know exactly what we're going to talk about.
Yeah.
We may bring in another person.
Yeah, we may bring in another person. That'll be fun.
Could be interesting.
Yep.
Maybe we'll get that guy who's washing the windows outside.
Why not?
Who knows?
Yeah.
In here. Thank you. Bye.
Bye.